Welcome to the lecture, which is called Biochemistry of Blood Cells and Plasma Proteins. It's actually going the opposite way because at first we will talk about plasma proteins and then about the blood cells. Here is the content where we see that we will talk about blood composition, blood plasma, about plasma proteins, functions of plasma proteins. I stress acute phase reactants and complement. The blood clotting we will only touch because main part of blood clotting is described in physiology. Then we will talk about metabolism of erythrocytes, white cells, and about platelets, but only from the uh, biochemical point of view. Blood composition. Blood is composed mainly by plasma and by elements where 55% is plasma, and of those 55%, 90% is water, and the remaining 10% are proteins, electrolytes, metabolic nutrients, waste products, hormones, and other signaling molecules. Of those elements, 98 to 99% are erythrocytes, and the remaining 1 to 2% are leukocytes and platelets. Plasma proteins, the concentration is 65 to 80 grams per liter, and there is less than 30 proteins. Of those, 35 to 50 grams is albumin, and 20 to 35 grams are serum globulins. Biosynthesis of plasma protein, majority is synthesized in liver, but for instance antibodies are synthesized by lymphocytes, and enterocytes are synthesizing also some of them, like apoprotein B48. Degradation, there are only two uh, degradation sites. First is hepatocytes, and the other one is mononuclear phagocytic system, which is capable to phagocyte complex of antigen antibody, hemoglobin, haptoglobin, and others. Plasma proteins, here we can see the same in graphical point of view, where we can see that about 50% of the plasma proteins is formed up by albumin, then there is immunoglobulin, transferrin, and others, C3 complement, and the remaining 10% are mainly apolipoproteins, which are part of the lipoproteins, and of course parts of the complement. Types of proteins, first one is albumin, the other one is are globulins, third one is fibrinogen, and what we have to remember is that actually the plasma proteins are differentiated by electrophoresis. We will talk about it in a while. And what is important is that under different conditions, like pathological one, pregnancy, and so on, the protein levels depart from the usual range. Here is what we see is the electrophoresis. Here is the positive charge of electrophoresis, negative charge electrode of electrophoresis. This is the agarose gel, and the agarose gel provides resistance to the moving proteins. So the proteins are dragged to the electrode by their charge, and the agarose is forming the resistance. So the proteins with the same charge and with different size mean that the smaller one is traveling faster than the bigger one because of the resistance of the agarose gel, but also the charge plays a role. So if we have a big molecule with a big charge, it can move as fast as small molecule with small charge. Alpha fractions you have to remember. So first one are albumins, then alpha 1 globulins, alpha 2 globulins, beta globulins. Sometimes they are differentiated to beta 1 and beta 2 globulins and gamma globulins, which are actually immunoglobulins. What is important is that albumin is more or less one molecule. Gamma globulins are molecules which are connected by their function, but alpha 1 globulins, alpha 2 globulins, beta globulins, they are actually traveling by their sizes and charges, which means they are not connected by their function at all, only by their physical properties. Functions of plasma proteins. First one is transport of substances, metabolites, hormones, waste products, anything. Number one of them is albumin, which is capable to transfer free fatty acids, bilirubin, calcium, drugs, hormones, copper. It means the molecules which are highly hydrophobic, and some of the cations. Transferrin is dedicated to transfer iron, ceruloplasmin, copper. Transcortin transfers cortisol, corticosterone, lipoproteins, lipids. Haptoglobin is catching and transferring free hemoglobin. Thyroid binding globulin, it is obvious, and retinol binding protein as well. The other function is osmotic regulation, because plasma proteins are colloidal, and they actually are providing the osmotic pressure, which helps to maintain normal blood volume and normal water content. If there is low level of albumin. Why albumin? Because it's 50% of all the plasma proteins. The osmotic pressure is lower, and therefore the blood is unable to take the water from surrounding tissue, leaving it there, and it's causing the edema. Other function is catalytic functions like lipases for removal of lipids from the blood, and protective function. Protective function is important because we have immunoglobulins which combine with foreign antigens and remove them, complement system which removes cellular antigens, enzyme inhibitors like alpha-1 antitrypsin, and they are actually trying to keep the um, function of, for instance, elastase only in the place where it is supposed to be. So if, for instance, some enzyme like some protease would go through the bloodstream, it will try to chop all the proteins into pieces which we don't want. We want those proteases to act only in one part, and some proteins increase during acute phase and protect body. We will talk about it in a while. The important thing of some plasma proteins is blood clotting, where many factors are involved, and we have thrombin, fibrinogen, and other clotting factors, and excess or deficiency leads to diseases. You already probably talked about it in physiology. Then, if there is blood clotting, there has to be anticoagulant activity, where plasmin breaks down the thrombin clot and dissolves the blood clotting. And buffering capacity, proteins help to maintain acid-base balance. However, you already talked about the basic one, which is the CO2 um, buffering system. Most of the proteins are synthesized in liver. Gamma globulins are not, or tissue plasmin activator is synthesized in enterocytes. They are synthesized as preproteins, and they go through post-translational modification. Almost all of them are glycoproteins except albumin, and they have a characteristic half-life in the circulation. For instance, albumin has approximately 19 days, but what we need to remember is that in some of the scientific papers I found 18 days, and in some of them are found 20, 21, 22. So it is at least 18 and at most 22 days. And most of them are polymorphic, immunoglobulins, transferrin, and so on. Then we have acute phase reactants, which levels actually change during acute inflammatory response. And the cause which starts the acute phase is destruction of cells, reversible cell damage, and metabolic activation of certain cells, mainly of cells of immune system. And acute phase reactants changes in infection, surgery or injury, and in cancer, because cancer cells are recognized usually as foreign cells. Here is the acute phase reactant response. So first we have the activated macrophage, which is sending interleukin 1, tissue necrosis factor, interleukin 6, and all those signaling molecules activate hepatocyte. So the hepatocyte starts to synthesize C-reactive protein, serum amyloid alpha, haptoglobin, fibrinogen, and others. But what is important is that there are some of the proteins which are synthesized in excess, like C-reactive proteins, its concentration changes 1000 times. Alpha-1 antitrypsin, fibrinogen, haptoglobin, C3, C4 parts of the complement system, serum amyloid A, and some of them are negative. First is albumin, because albumin serves as the source of amino acids. So the synthesis of amino acids is decreased and dose is increased. Then we have transferrin. The transferrin is part of the thing which is called quest for iron. It means in the case of bacterial infection, the bacteria needs iron for synthesizing cytochromes, which are part of the bacterial electron chain, and if the concentration of available iron is decreased, the conversion of energy to ATP in bacteria is also decreased. Then we have transcortin. Why transcortin? Because transcortin binds cortisols, then retinol binding proteins. The importance of positive acute phase reactants is the components of immune response, the C-reactive proteins, complement components, protection against collateral tissue damage, which is haptoglobin, hemopexin, ferritin, ceruloplasmin, and protease inhibitors, which are trying to protect the tissues against the uh, so-called escaped proteases. Now, of course, if there is some inflammation, there is high level of waste products. So hemoglobin, hemopexin, and serum amyloid are transporting waste products. And then there is the activation of coagulation factors like fibrinogen, plasminogen, and so on. And coagulation plays important role in immune response by immobilizing or partially immobilizing the bacteria. The importance of negative APRs, of course, the criterion for determining inflammation, transcortin B, because corticosteroids are actually decreasing the negative the immune response. So if there is higher concentration of cortisol, the immune response out of the site is decreased. And it's the criterion also for protein synthesis in the liver. First one is albumin, where concentration of albumin is 45 grams per liter. Functions, of course, the oncotic pressure, and if there is a lower level than 20 grams per liter of albumin, it leads to edema. And transport steroid hormones, free fatty acids, bilirubin, drugs, and the ions. What is somehow important is the transport of copper. Alpha-1 antitrypsin, which is main globulin of alpha-1 fraction, is synthesized in the liver. It's a glycoprotein. It is synthesized also by macrophages, highly polymorphous, and it's the main plasma inhibitor of serine proteases. And during acute phase increases, so it's the inhibiting the degradation of connective tissue by elastase. And there is one thing, deficiency in alpha-1 antitrypsin is actually uh, leading to proteolytic lung damage. Why? Because in lung there are also macrophages and neutrophils. The neutrophils are releasing the serine proteases as a defense system against, for instance, bacteria, and those proteases are actually inhibited by the alpha-1 antitrypsin, but if the alpha-1 antitrypsin is not in sufficient concentration, the proteases are capable to destroy also things which are not meant to be able. Transferrin is beta-globulin, binds iron, binds two molecules of Fe2 plus ions, and it's normally saturated about 1/3. Transferrin levels are decreased in liver disease, chronic infection, nephrosis, and inherited disease congenital transferrinemia, and increased serum transferrin levels occur during uh, increased transferrin synthesis caused as a result of iron deficiency anemia. What does it mean? If there is not enough iron, for instance, in the nutrition of iron can't be digested in sufficient levels, the liver starts to increase transferrin level in order to keep all the iron it is capable. And also this is marker of some of the intestine diseases like Crohn's diseases or celiac. Here is why actually the transferrin is transferring the ions. So transferrin with bound iron is reacting with the receptors on the plasma membrane, and then by clustering coated way, the clustering coated vesicle or endosome is formed. Then the endosome is changing its internal pH by deploying the proton pump, and the pH inside the endosome is decreasing. Once the pH is low enough, the iron is removed from the transferrin and is transferred by specific transporters to the cell, and the endosome without iron but with the transferrin receptor is then joining the plasma membrane and releasing the transferrin without iron. So we can see that this is quite complex thing. Ferritin, on the other hand, is intracellular protein, and only a really tiny portion is in plasma. And in comparison with transferrin, where transferrin binds to Fe2 plus atoms, the ferritin bounds about 3,000 to 4,500 ions of Fe3 plus, and function is storage. So it stores the iron, and the disease is primary hematochromatosis, where genetic disorder is characterized by increased absorption of iron from the intestine. Then the accumulated iron damages organs like liver, skin, heart, pancreas, and the body tries to fight against the high level of iron by increasing the level of ferritin. Ceruloplasmin, the concentration is 300 milligrams per liter and it carries about 90% of copper in plasma. So one molecule binds 6 atoms of copper, but the bond is stronger, so the ceruloplasmin binds copper more tightly than albumin. So albumin carries about 10% of plasma protein, but the copper is easier to release and it plays also important role in oxidation of Fe2 plus to Fe3 plus. Haptoglobin is alpha-2 globulin, tetramer formed of 2 alpha and 2 beta chains. It's in 3 polymorphic forms, and what is important is that it binds free hemoglobin and it delivers it to reticuloendothelial cells. The other advantage is that hemoglobin haptoglobin complex is so large that it can't pass through glomerulus, therefore keeping the iron in the body. The problem is that free hemoglobin is capable to pass through glomerulus and in the tubules it quite often precipitates, and of course precipitation in um, kidney tubules leads to kidney damage. Here is one of the important thing, and it's plasma proteins as antioxidants, where all those transferrin, ferritin, haptoglobin, hemopyxin act as antioxidants by removing the bivalent Fe2 plus iron and prevents the Finton reaction. The Finton reaction is described here where Fe2 plus reacts with hydrogen peroxide forming Fe3 plus hydroxide ion and hydroxide radical. This hydroxide radical is highly reactive, attracts with almost everything, and of course it destroys the target molecule. C-reactive protein belongs to beta-2 globulin, which rises in during inflammation, so it's acute phase reactant, and it binds to phosphocholine expressed on the surface of dead or dying cells and sometimes of bacteria. So it's part of the inherited immunity, so it means while the C-reactive protein is capable to find the bacteria, it's capable to react with the bacteria. And plasma concentration of C-reactive protein increases within 2 hours. Fibrinogen is glycoprotein, beta-2 globulin. The concentration is 1.5 to 4.5 grams, and it's component of the coagulation cascade. It is acute phase reactant, so it increases during acute inflammation. And the last one are immunoglobulins, antibodies produced by B cells and react specifically with antigenic determinants. Plasma enzymes, specific enzymes like cholinesterase, plasma superoxidismutase, lecithin acyltransferase, and serine proteases, which are in form of inactive zymogens, and those are mainly the parts of coagulation factors or coagulation cascade and complement system cascade. Here are some plasma proteins like disease markers, alanine aminotransferase and aspartate transaminase, also ALP. Those three actually shouldn't occur in the blood because usually those are markers of some damaged cells. And here the same applies. So here we have lactate dehydrogenase and gamma glutamyl transferase. Again, those proteins are supposed to be inside the cells. Now we are getting to complement system, which consists of more than 30 soluble proteins circulating in the blood, and it's activated in three ways. First one is classical complement pathway, then the lectin complement pathway, and alternative complement pathway. What is important is that actually here we have microbe, and there are several ways how actually the microbe can be eliminated by complement system. First one is alternative pathway, which is phylogenetically oldest, and it interacts with C3b part of the complement, which is reacting directly with the surface of the microbe, marking it for other process, where one of them can be phagocytosis and the other one is formation of membrane attack complex. Then we have classical pathway where the microbe is or bacteria is recognized by antibodies. The antibodies are opsonizing or marking the bacterium for destruction, again reacting with C3b part of the complement, and again two ways, phagocytosis or formation of membrane attack. And the third one is that mannose binding lectin, which is present in blood, is directly reacting with the surface of the bacteria, marking it in the same way as antibody for destruction, and then again those two pathways. The marking for phagocytosis is quite obvious, but what is important is here is the membrane attack complex. Membrane attack complex is formed from several proteins, and all this complex is then drilling a hole in the bacterial membrane, and once the membrane is actually destroyed or uh, there is a hole made in it, the electrochemical potential is discharged, so the bacteria is unable to form ATP, or their ability to form ATP is highly decreased, and then it's destroyed. Here is the same like what was before, but it's only in different details. Again, what is important is the classical pathway, lectin pathway, alternative pathway, and then the activation of membrane attack complex or marking for the phagocytosis. The third way, where we can see that all three pathways are actually connected to C3 convertase, where C3 convertase is converted to C3b and C3a, which is part of the C3b, and it actually is serving as the signaling molecule for sending the information. The inflammation is going on, there is some attack. This is one of the immune response, the non-specific immune response, and this is actually uh, part of the plasma proteins. So now we have erythrocytes, where erythrocytes, they have the um, typical shape. We have 5 million per milliliter in men's blood and 4.6 million in women's blood. So the volume is 90 femtoliters and the concentration of heme is 330 grams per liter. The other thing is, from our point of view, which is specific for erythrocytes, is cytoskeleton. Cytoskeleton is made by special molecules which are called spectrin, and those spectrin molecules are attached to uh, anchors which are called ankyrin. So spectrin is bound to ankyrin and is forming this mesh-like pattern, uh, hexagonal mesh-like pattern, and the thing is also important that it gives erythrocytes really easy ability to change the shape and take the original shape immediately after the change. So they are highly flexible. Here we have erythrocyte metabolism. Erythrocyte metabolism is quite simple. It's just pentose phosphate pathway and um, glycol glycolysis. So again, glucose, fructose, and anything, and there are several things which actually have to be mentioned. First one is cytochrome B6 reductase, where NADH is not forming only lactate, but it's reducing the cytochrome B4, B5, forming cytochrome um, B5 reduced, and this reduced cytochrome B5 is able to reduce Fe3 hemoglobin back to functioning Fe2 hemoglobin. Other thing which is important is bisphosphoglycerate mutase or Rappaport-Luberin shunt, where 1,3-bisphosphoglycerate is forming 2,3-bisphosphoglycerate, which is actually one of the allosteric activators or inhibitors of hemoglobin. And here is also the pentose phosphate pathway, which is used mainly for reducing the glutathione. So the thing is that hemoglobin autooxidation is occurring spontaneously where oxygen binds to Fe2 plus, but what can happen is that the oxygen is not released in form of Fe2 plus, but in form of uh, superoxide anion and Fe3 plus. And Fe3 plus is unable to bind oxygen, and usually 3% of the hemoglobin undergoes oxidation every day. So we have to actually reduce it, and it's reduced by the cytochrome B5. Now we have glutathione synthesis. Glutathione is tripeptide, and this is form of reduced glutathione. The reduced glutathione has SE/SH group, but once it's oxidized, two glutathiones are bound by um, disulfur bond and forming dimer. And what is important is that actually the pentose phosphate pathway generates the reduced glutathione, and it eliminates hydrogen peroxide or superoxide, which is formed in erythrocyte. And what happens if there is deficiency in glucose 6-phosphate dehydrogenase? It's causing hemolytic anemia, where the decreased production of NADPH is forming higher level of hemoglobin oxidation and Heinz bodies formation where membrane lipid peroxidation and hemolysis. So hemolytic crises are invoked also by drugs like primaquine, sulfonamide antibiotics, and some food. Broad beans are the most famous one. And this is the most common enzyme deficiency disease in the world. It's affecting approximately 100 million people. Here is the marker of degradation of the heme. It's just for you to remember. You already heard about it in the first semester. So here you have heme, bilirubin albumin, and bilirubin deglucuronide, where bilirubin is excreted. Here is the iron cycle reminding, which is showing how actually the body is trying to keep as much iron as possible. And now we are going to white blood cells. So you can see we have the neutrophil, eosinophil, basophil, lymphocytes like B cells, T cells, and natural killer cells, monocytes, and they are forming different resident macrophages, free macrophages, and dendritic cells. So those are the white blood cells. Yes, it was taken from Wikipedia, but I have controlled the things are correct. So white blood cells, we have phagocyting cells, basophils and mast cells, and lymphocytes. The lymphocytes we will mention really fast because you will hear about this later. First one are regulation. Many functions of leukocytes are regulated by monomeric GTP binding proteins, which is activating the NADPH oxidase, chemotaxis, phagocytosis, and fusion of phagosome with granules. RHO and RAC are able to modulate the assembly of actin filaments and therefore increasing the speed, rate, movability of those white blood cells. Here we have phagocyting cells where the first ones are granulocytes, neutrophils, eosinophils, basophils, then monocytes and macrophages, which rise by differentiation of monocytes in tissues. Now, degradation. First one is activation of NADPH oxidase, then production of um, nitrogen monoxide by nitric oxide synthase, and fusion of phagosome with lysosome of the phagocytic cell, which contains hydrolytic enzymes and usually acidic pH. First enzyme which is involved in the immune response is NADPH oxidase. It's complex of neutrophils, eosinophils, monocytes, macrophages, and the reaction is that NADPH reacts with two molecules of oxygen, forming two molecules of superoxide anion, and two molecules of superoxide anion react with two H plus ions, forming oxygen and hydrogen peroxide. And hydrogen peroxide can damage bacteria directly or after conversion to hydroxide radical. So in this case, the Finton reaction is desired, so because it is aimed to destroy the pathogen. Here is the NAD. pH oxidase activation is by association of the components localized in cytosol. So we can see microbials, inflammatory mediators, and cytosols, and all um, are actually assembling the activated NADPH oxidase, and again the NADPH is then transferred via FID and cytochrome to oxygen. Here it is a little bit simplified, so outside, inside the activated NADPH oxidase is actually producing the superoxide anion, and in case of phagosome is actually increasing the level of reactive oxygen species inside the phagosome. And of course, if there is high concentration of hydrogen peroxide, the bacteria or pathogens are definitely not happy. Myeloperoxidase is present in granules of neutrophils and monocytes, but not macrophages. And what does it do? The myeloperoxidase, it takes a portion of hydrogen peroxide and it is used to oxidize to chloride ions to hypochloric acid, and hypochloric acid is highly reactive and it provides also toxic chlorine gas. The other thing is, you know, hypochloric acid or hypochlorites. It is used as common bleach under the name Savo or Domestos and so on, and it's actually dissolved uh, hypochlorite acid salts. Also, hypochloric acids react with superoxide anion and is forming OH2 radical. What is chronic granulomatous disease? It is caused by deficiency of one of the NADPH oxidase subunits, so superoxide and other reactive species are not produced, and usually the person is unable to fight the pathogens. So usually there is the infection by Aspergillus, which is causing pneumonia, or Burkholderia, which is causing pneumonia, and so on. And usually, if person suffers chronic granulomatous disease, it is important to use antibiotics and antifungal agents. Here is other thing which is produced, and it's nitric oxide, and it's produced by inducible nitric oxide synthase, which takes arginine, and it's reducing this part of the arginine to NaOH, forming citrulline and releasing the nitric oxide production. And this can kill bacteria directly or indirectly by reaction with superoxide anion and generating peroxynitrite, and it attacks iron sulfate proteins, mainly the SH groups, which inactivates the enzymes. NADPH oxidase and NO. NADPH oxidase is effective mainly in degradation of extracellular pathogens, Salmonella, Streptococcus, Staphylococcus, and nitrogen oxide serves mainly to kill intracellular parasites, Listeria, Brucella, Candida albicans, and also in macrophages. Granulocytes, all of them are phagocytes, usually contain several types of granules, usually primary granules, which are participating in phagocytosis, and secondary granules, which are releasing cytotoxic and immune response mediators like defensins, catepsins, and others, and also other proteases. And after the phagocytosis is accomplished, the respiratory burst occurs. What is respiratory burst? It is actually the moment where the cell needs a lot of oxygen in order to fight the pathogen. Number one are neutrophils, which are actually about half of the white blood cells. They have to be activated and interact mostly with opsonized cells. What are opsonized cells? Those are cells where on the surface is or attached some antibodies, or, as we mentioned before, parts of the complement C3b part, which is marking for digestion. And neutrophils also mediate other immune response by sending eicosanoids and cytokines to actually activate the other parts of the immune system. The main target of neutrophils are bacteria. They contain myeloperoxidase, we talked about it, which is producing the hypochloric acid. Of course, the part is lysosome, which is lysozyme, which is cleaving the glycosidic bond in peptidoglycan, which is on the surface of bacterial cell walls. Then they are releasing defensins, which are cationic peptides, a lot of arginine, and they interact with anionic lipids on bacterial membrane and make spore in it, and also inhibit synthesis of DNA and proteins because it binds to the DNA. And the last one is they can actually attach to the flagella of bacteria, making them less mobile. And of course, they are releasing hydrolases like elastase and other serine protease, which can damage bacteria and cleave virulence factor. But the unwanted problem is harm of the host tissues and where the alpha 1 antitrypsin goes into the playing role. Eosinophiles usually they are fighting against eukaryotic parasites, so increased level of eosinophiles is one of the marker of malaria. Again, the reactive oxygen species production, they contain eosinophil peroxidase and not myeloperoxidase, but it prefers bromide substrate generating hyperbromic acid, and it's even more reactive and more aggressive than hypochloric acid. Again, they are releasing proteases, and the granules contain major basic protein which is cytotoxic to parasites, and it's also marker for us because they are basic, so they can be marked by eosinophil eosine, which is color marking the basic parts of the molecule, and they are capable of releasing histamine. Here what we can see is eosinophiles which are capable to activate T cells, and major basic protein can activate also mast cell. And what are secreting is major basic proteins, others cytokines, chemokines, lipid mediators like leukotrienes, platelet activating factors, and it can be stimulated by allergens, tumors, and others, and also by interacting with some of the pathogens. Last one are basophils. They are mainly immune response mediators, and they are releasing histamine and serotonin. They contain immunoglobulin E receptors, and once activate, they degranulate. And because if they degranulate too fast, they are responsible for allergic symptoms, and they activate synthesis of eicosanoids, and leukotrienes are bronchoconstrictors which also stimulate chemotaxis and leukocyte activation. Here is some graph what the basophils are capable to activate, and we can see that there are a lot of other things which are activated by basophils, by histamine and other signaling molecule. What I can to tell, this is not going to be part of the exam, it's just for your illustration how complex the activation by basophils is. Here is what is histamine. It's produced by histidine decarboxylation. Histidine decarboxylation, decarboxylase removes the COO minus forming histamine, and it causes vasodilatation and bronchoconstriction, which helps to eliminate parasites, coughing it out, peristaltics, and enhance production of mucus, which is drawing and easily removing the parasites from instance in lungs or atopy. If immunoglobulins E are recognized allergens like pollen, food, and they are produced and bind to immunoglobulin E receptors of basophils. At next exposure, they can lead to release of histamine and heparin and synthesis of eicosanoids. Local symptoms, allergic rhinitis, asthma, conjunctivitis, but if the allergen enters bloodstream, it is causing massive degranulation of basophils, and it can lead to decrease in blood pressure, pulmonary edema, and anaphylactic shock, and the treatments are antihistamines which block histamine receptors. Here is mentioning the monocytes macrophages, and the um, main enzyme activity they are inhibiting are cathepsins and others. So the cathepsins are the main proteins which are actually released by monocytes and macrophages. Then we have fixed leukocytes. You already heard about them: histiocytes, dendritic cells, mast cells, microglia, and Kupffer cells, which are leukocytes embedded in the tissues where they are waiting for the infections to appear, and they are ready to fight. Here is one thing which is important, is that actually each cell is capable to fight viruses. If the viruses enter the cell, they are usually marked by double-stranded RNA, and the double-stranded RNA is then interacting with other parts of the signaling pathways in the molecules, and it can lead in massive infestation by viruses to activation of caspases and apoptosis, and also by other NF-kappa B and other pathways. It can lead to forming endosome, which is swallowing, targeting the double-stranded RNA. Also, there are endosomes which are capable to destroy single-stranded RNA, double-stranded RNA, and also what is important is that also peroxisome plays important role in eliminating the viruses. But what is important is that all those signals are ending in the nucleus when they activate interferon alpha B genes, tissue necrosis factor alpha, interleukin 6 genes, inflammatory cytokines, and all those which are actually started to be produced and released. So the the cell is telling, please, I am infected, I am fighting, activate yourself, get ready, and send us some help or someone to destroy us. Here is the same, only in the surrounding cell, where the interferon is or acting autocrine or paracrine, where it is interacting with interferon receptor. The interferon receptor is activating STAT1, STAT2 pathway, and the pathway is actually activating the enzymes which are responsible for synthesis of antiviral effectors like viperin, tetherine, and others, and positive regulators which are sending the signal to the surrounding tissues and to the body. Here the infection is going on. Please, those two things you don't need to remember, only that each cell is capable to defend itself and also to sense that it is infected by virus and send the signal, I am infected, activate the immune system, help us. Blood coagulation, now we are talking about platelets. They are not nucleated, they are granulated. They contain electron dense granules, and those granules contain calcium as a signaling molecule. Adenosine diphosphate is a signaling molecule, also adenosine triphosphate and serotonin. And granule which contains heparin antagonist, platelet derived growth factor, thromboglobulin, fibrinogen, von Willebrand factor, and other clotting factors, and some lysosomal granules which contain hydrolytic enzymes. Those are the granules which are present in platelets, and first thing is they form blood clots, act as vasoconstrictors, and participate in defense against infection. So they suppress the growth of Plasmodium falciparum, which is actually attached to the Plasmodium surface and doesn't allow the plasmodium to be as easy to move, and also it destroys the surface um, plasma membrane. They are capable to generate superoxide, anions, and hydrogen peroxide, which and they also can synergize with aggregatory stimuli. And what they contain is thromboxane A synthase that catalyzes the conversion of prostaglandin H2 to thromboxane A2. And thromboxane A2 promotes platelet aggregation and vasoconstriction. Now the platelets which are important for us are platelet activating factor and platelet derived growth factor. Platelet activating factor has this structure and acts mainly juxtacrine and paracrine signaling, promotes platelet aggregation, and induces activation of leukocytes, adhesion, chemotaxis, cytokine production, and also causes vasodilation and bronchoconstriction, and mediates interplay between thrombic and inflammatory cascades. It can be also produced by endothelial cells, monocytes, granulocytes. And what is actually the platelet activating factor? It is a phospholipid. So platelet activating factor is phospholipid, which is actually starting the blood coagulation, the immediate response to injury or infection, mainly to the injury. The other one is platelet-derived growth factor, which is dimeric protein in 3 isoforms. The receptors are tyrosine kinases, leukocytes, and so on, and it actually activates proliferation, chemotaxis, and cytoskeletal rearrangement and differentiation of certain types of cells. And this is what is responsible in wound healing, capillary formation. So this is the signaling molecule which is telling the body, please repair this part. And here is the literature. Thank you very much for your attention, and in case of any questions, please use the Moodle discussion, and my colleagues will be happy to answer all your questions.